Hello Nutri subscribers, welcome back to Nutriverse Academy. So in previous episode we explained about nutrigenomics. So today we are going to explain about tools of nutrigenomics. So generally in this chapter you're going to learn about omics technology. So where did you listen this omics technology? I was explained in the last few minutes of the system biology approach. In there you have listened about this omic technology, how this omic technology were introduced or how this is used in nutrigenomics. Today we are going to listen about. So generally omics technology, we will have 4 omics technology. Those are genomics, proteomics, and next is metabolomics, and next is transcriptomics. So these are the omics technology that was introduced in the nutrigenomics. So these are the tools which were introduced in the nutrigenomics. So generally before knowing this genomics, proteomics, metabolomics and transcriptomics, as you already identified that generally these genomics have the two definition. Omics is the technology and gene means genes. That means here we are introducing gene technology. Okay, and in proteomics here protein, and in same here metabolomics means metabolism of the body, right? And in transcriptomics RNA, mRNA, right? So these are the technologies that are introduced in the genes, proteins, metabolisms, and in mRNA, that is transcription we will learn in the genetics, right? So for that we will learn genomics, proteomics and metabolomics and transcriptomics. So in prior knowing of these technology, you need to know a pathway of the analysis. So how these analyses were done. So how these analyses were done, you have to know. So generally if we go to the genomics, generally what we will do, first the researcher will analyze particular gene, and then they will give they will identify the gene expression, what that gene is expressing, and the next is they will know if there is any variation. Obviously there will be a variation, right? For each code there will be a variation, for that gene variation, and how this gene variation is interacting with the diet. So there is a dietary change. So after knowing that all, they will implement the nutritional strategies. So intervention of these strategies will go. So that will helps to promote healthy care, right? So that will help to promote healthy care. Once again I'll write the pathway of the analysis, how the pathway of the analysis will be given. So it is not only goes with the genomics but also for the proteomics, metabolomics and mRNA. So the pathway of the analysis, generally researcher will first what he will do, he will analyze particular gene. And then how he will do, he will analyze particular gene, and then he will encode gene expression and variation. Okay, gene. And then how we will do, after encoding those all, he will see gene diet interaction, and after that what he will do, intervention of the strategies. So intervention of strategies, after that we will promote the health care. So for this one example I'll tell you. So if a person is suffering with lactose intolerance, that means he can't drink or he can't eat any dairy or dairy products, any milk or milk products, or anything dairy that is linked with dairy products or dairy. Okay, so what the researcher will do, researcher matlab anyone either the lab technician or either the dietitian or either the nutritionist or either the RD. First how he will do, he will or she will give the identify the particular gene, whether he is expressing or he showing any variation to particular diet, and then after knowing that he will intervene the strategy and he will say whether you have lactose intolerance or not. So it is not only through the gene but also through the biochemical parameters also. In prior of this knowing, then he will include this technology. So that is the main analysis of the tools of nutrigenomics. So this is the omics technology. So where the same analysis will goes to the genomics, proteomics and metabolomics and transcriptomics. So generally in question paper how we will get means, so either for long answer either they will ask write about genomics and proteomics, or proteomics or and metabolomics, or transcriptomics and genomics. We don't know how they will ask. If they will ask either 2 means, then you can write for 3 marks answer for this and 3 marks answer for this. Total will get 6 marks, right? So we will get 6 marks easily. If they will only give proteomics, then you need to include definition, and next applications, interventions, and also some type of technology which is included with genomics, proteomics, metabolomics and transcriptomics. Okay, so for each and one I have made the notes, so you can get in the description, and you can also join in the WhatsApp group, okay, which I will provide in the description itself.